final but not least talk this afternoon is from Hind Mandoor. And the talk is on genetic variation in transpiration response to vapor pressure, deficits in 2 faba bean cultivars using different methods. Cultivars are Masterpiece and Robin Hood. Are you going to tell us why it's called Robin Hood? And I'll hand over to you anyway.
Thank you. Have you got my screen? Welcome. Hi everyone. I'm Hind Mandoor, PhD student at Lancaster University, UK. My final year. I'm supervised by Professors Indod and Elizabeth Silva, and funded by both the Egyptian and British governments via Newton Musharrafa Fund. In this session, I'm going to accompany you in a very short journey on a methodological study on genetic variation in transpiration response to VBD in faba bean using different methodologies. As most of you know, faba bean is one of the most important cool season crops in many parts of the world. It can be utilized as a food or feed because of its high nutritional value, high protein content, carbohydrates, minerals, etc., which makes it one of the best solutions to malnutrition in the developing countries. Also, like most other legumes, it is symbiotic with nodule-forming bacteria that can fix nitrogen from the atmosphere and consequently save fertilizers to the upcoming crops. However, this cute crop faces so many troubles nowadays because of the biological limitations of the traditional cultivars, the effects of biotic and abiotic stresses, and its sensitivity to water shortage, which is quite sensible nowadays because of the climate change. So the ideal solution in this situation would be to improve the existing varieties for water use efficiency, to make good use of water and give high productivity. My main interest here is vapor pressure deficit as one of abiotic stresses that somehow helps in drought tolerance. So VBD is the difference between vapor pressure in the air and the maximum vapor pressure the air can hold when it is saturated. In other words, it is a difference between vapor pressure in the leaf and the surrounding atmosphere. It is a combination effect between temperature and relative humidity. Normally, high VBD occurs as a consequence of elevated temperature when vapor pressure in the air is saturated, and this can create a kind of water deficit around the leaf when the rates of water losses exceeded those of water uptake. So logic-wise, the plant should increase its transpiration under high VBD to compensate the water uptake, and because also transpiration works as a cooling system, but sometimes this is not the case. Sometimes the plant decreases its transpiration despite being exposed to a high VBD, and this is a very promising response because it works as a water conservation strategy. It can save water to the most critical periods like flowering and grain filling, which are normally considered with high temperature in faba bean growing season. On the other hand, it doesn't have any negative impact on the final output, based on some simulation studies conducted around the world, where limited transpiration response under high VBD caused a penalty reached to 75% in wheat productivity in some locations in the United States. So this is VBD, and that's how a plant responds to VBD. The question now is how we can measure transpiration response to VBD. Of course, the measurement should be conducted precisely and rapidly, and if we can make it easily, it would be perfect as well. So it could be measured under naturally fluctuating VBD in potted plants in the glass house, either gravimetrically from the difference in pot weight between successive measurements, or with the infrared gas analyzer, which controls VBD around the leaf. And or alternatively, it could be measured in the whole plant gas exchange chamber, which provides more control of VBD around the whole shoot. This system was invented by Ivan Jaigoy in Lancaster Environment System Center 2018. It combines between the previous mentioned one, and its advantage over them is VBD is achieved under almost stable temperature, unlike the balance and the gas analyzer, where VBD varies between temperature and relative humidity, and we aren't quite certain which one affects transpiration most. So I tested the C systems with Master Beats and Robin Hood, commercial faba bean genotypes under 7 VBD levels within the range of 1.5 to 3.5 kilopascals, more or less, to see to what extent the response would be consistent over the two systems. We hypothesized that the two systems are different, and the two cultivars are genetically different as well. So after 7 months in this experiment, I think I was lucky because both cultivars revealed segmented linear transpiration model over the C systems with a breakpoint of 2.4 and 2.9 for Robin Hood and Master Beast respectively. And after repeating these experiments for four times, I think, now I have a solid conclusion, and I can say that these three systems are quite consistent, and using any of them is based on the size of the population or the time, etc. Also, the response have been confirmed in different time of the year and different time of the day. So I can say this trait is stable, genetically stable, and we can select for it directly. We didn't get any significant difference between July and October and between the 3 different times of the day. Also, the two genotypes have revealed genotypic difference in their transpiration response to VBD. Moreover, they differ in their slopes, the first and second slope. Also, an interesting observation here that the genotypes that revealed lower peak point, which is Robin Hood, has higher slope 1 value. This means that this genotypes is quite suitable for dry areas, because it maximizes its transpiration and its gas exchange under low VBD, and as VBD increases, it shifts to water conservation strategy. Unlike Master Beast, which would be a bit tricky to adopt it in dry areas, it might be quite suitable to well-watered conditions. So to conclude, the three systems are quite consistent, and the choice of phenotyping system will be determined by the size and the genetic diversity of the population. Both genotypes revealed segmented transpiration model in their transpiration. This trait is stable and independent from either the time of the day or the time of the year. And finally, the two genotypes are, the two cultivars are genetically different. For future research or future plans, since this is the first report in faba bean, further experiments were needed to figure out why this response happens, or what are the physiological mechanisms beyond this response. We started by measuring hydraulic conductance at different plant parts and abscisic acid from different plant tissues, and the preliminary data are quite interesting. It will be published soon. Also, I'll have to locate the genes responsible for the trait along the faba bean chromosome via K2L analysis. Finally, I'd like to thank Ian, my lovely supervisor, for his in-depth guidance, support, sometimes we fight. Also, I'd like to thank my funding body for their financial and psychological support. Also, thanks to the British Council for always posting stories about my progress here at Lancaster University on their social media platforms. And thank you all for your attention, and your questions are most welcome. Also, my email is here in case you want to understand in details how we conducted these measurements. Thank you.
Thank you very, very much. And again, Hend, you've kept completely to time, which is perfect.
Thank you.
We now come to the end of the session, and I thank all the speakers once again. We have to move across to another Zoom site for